So this brings us to the last presentation of the morning session. Afterwards, snacks and coffee will be served outside of the seminar rooms. And the last presentation in this morning session will be given by Cintia Gómez, scientist at Demeetra, and she will talk about unveiling the complementary benefits of Cas-CLOVER dimeric nuclease and harbor-in license-free transposase. Dr. Cintia Gómez is an accomplished biotechnologist specializing in cell line engineering. With over 12 years of experience, she has worked extensively with various types of cells, including induced pluripotent and hematopoietic stem cells, and at Demeetra, CHO and TEC293 cells. Dr. Gómez has hands-on expertise with non-viral delivery methods, such as extracellular vesicles, transposon systems, as well as viral delivery systems like AAVs. At Demeetra, Dr. Gómez focuses on cell line development using advanced technologies, including Cas-CLOVER, the clean alternative to CRISPR-Cas9, and transposase systems to enhance applications in bioproduction and gene therapy. So, we're looking forward to your talk.
Thank you so much. Good morning, everyone. You can hear me well? So, thank you so much for the opportunity to present our technology in this conference. I'm Cintia Gómez. I'm scientist cell line development. And the title of this presentation is Unveiling the Complementary Benefits of Cas-CLOVER dimeric nuclease and harboring license-free transposase. So before showing the technology and our results, I would like briefly talk about Demeetra. Demeetra is located in Lexington, in Kentucky. Demeetra is a gene editing company, and it was founded in 2019, and 80% of this company is hands-on research. As you can see, we are an interdisciplinary team with extensive experience in different fields, and together we focus on optimizing the gene editing technologies for commercial application. So, for example, we established the CHO cell line development platforms, and I will show during this presentation. Together with the optimized protocols, we like to share our knowledge with our clients in order to improve the results. And in addition, we have an innovative licensing with freedom to operate, and I will talk in more details in the end of this presentation. So Demeetra have two main technologies. The first is the harboring transposon system. This system consists in two components. One is the transposons, in which we can add one or multiple transgenes. This is an advantage for bioprocessing fields. So the cargo of this transposon is flanked by an ITR sequence, which together with the transposase will help for the integrations in the host genome. These integrations will be inside a specific rich in TT-AAVR pairs, which are more frequent in no-coding regions. And that is an advantage for cells and gene therapy. So the second technology is the Cas-CLOVER target nuclease. Here you can see Cas-CLOVER have two parts. You can see that we have the left and we have the right, and each of them has a nuclease inactivating the Cas9 protein. When we complex together with the guide RNA, this protein will receive the directions to target the specific genes. However, this protein is not able to cleave. Instead, there is together an endonuclease called CLOVE51. And when these two parts arrive in this specific site together, what is happening is that the endonuclease will dimerizate and become active, giving for the Cas-CLOVER an exceptional precision to cleave the specific site. So Cas-CLOVER has a high efficiency target dimeric nuclease. We can observe larger deletions, higher knock-in efficiency, and lowest of target because in part of this exceptional precision. When we compare with the conventional Cas9, we observe a difference, such as we observe more small indels in Cas9 compared with Cas-CLOVER, and frequency of target events is observed in Cas9. Cas-CLOVER is a fusioning protein, and as part of this group of proteins, there is challenge such as express and purify these proteins. So, however, you can see here Demeetra was able to produce this recombinant protein, and we have an in vitro assay in which we can evaluate the mechanisms of cutting and the kinetics of Cas-CLOVER. So we are using an amplicon with a specific target, and we know that Cas-CLOVER will cut between the guide RNAs, the left and right. And Cas9 will cut using just one guide RNA close of the PAM sequence. So when we evaluate the type of cut regarding Cas-CLOVER, we observe that we have more stick-ended cut compared with Cas9 that is more blunt-ended. The blunt-ended is more associated with in a specific chromosomal rearrangement compared with the overhand-ended that is more observed, less translocations. So now moving on for the cells, we evaluate the editing efficiency using Cas-CLOVERs, and also we compare with Cas9. We use CHO cells and this glutamine synthetase target that is a top target for cell development. And here you can see that we use different versions of Cas-CLOVER. The first is a mRNA Cas-CLOVER, and the second is a recombinant protein Cas-CLOVER. So in these comparisons, in the first experiment, we include the Cas-CLOVER versus Cas9, and also to show that Cas-CLOVER can work with different companies, we evaluated the single guide RNA synthesized from IDT or Centego, and also we compare with the two parts of guide RNA. You can see, to briefly explain that, we mimic the nature in which we use the guide RNAs pre-complex with the tracers, and then we incubate with the mRNA. And what we observe regarding the editing efficiency, which we evaluate using the T7 assay, we observe a comparable editing efficiency when we compare these guide RNAs, around 75%, and was higher when we compare with Cas9. Another experiment was using the recombinant protein, and we observed comparable editing efficiency when we compare Cas-CLOVER with Cas9. But here we would like to highlight that using the protein, we have some advantage. For example, we know that using the plasmid, the expressions of the Cas-CLOVER will depend on the reporter. And also the translations of this Cas-CLOVER will depend on the machinery of the cells. Using the protein, we have the final product. It's ready to go and edit the genome. And the second advantage is that this protein can be used in different types of cells without any modification. So during these years, Demeetra was working with different types of cells, and we test this technology, the Cas-CLOVER, in these different types of cells. So starting first, we have here the work that we did in plants. For example, in the tobacco plants, we knock out the PDS genes, and we correlate the editing with the phenotype of these plants, which change. We also work with yeast. We knock out the genes AD, and we observe that the edited colonies change the color, the red phenotype observed in this image. And in addition, we work with E. coli. What we did was establish an E. coli date screening assay, which basically help us evaluate the activity of Cas-CLOVERs in different conditions in a high throughput manner. And finally, we included the mammalian cell lines in which we work with CHO cells, HEX cells, and recently we include the T cells for cells and gene therapy. This is the last comparison between Cas-CLOVER and Cas9, and what we did was evaluate the knock-in efficiency between them. And we use an integration site of 7 kb, but our company has experience working up to 20 kb of integration. And in this specific experiment, we use HEX cells, You can see here that we evaluate the knock-in efficiency based on GFP expression. And using cytometry into the alive populations, we evaluate the GFP expressions, getting around 10% for the Cas-CLOVER versus 2% in Cas9. Because we also use puromycin as a resistant marker, we enrich the populations, achieving 70% for Cas-CLOVER conditions versus 32% for Cas9. So summarizing, you can see in this table what we have is the comparison between Cas-CLOVER and Cas9, and what we observe is regarding of target and translocations, we observe 0 or very few quantities of off-target. The activity was comparable in terms of knockout for Cas9 and higher in knock-in efficiency. The index, as I mentioned, was larger in Cas-CLOVER, small in Cas9, and highlighting regarding the IPA licenses, we have a commercial freedom to operate in a single simple license for Cas-CLOVER, compared with Cas9 that show major complexity. So this is the first cell line that we developed. This is the commercial clean cut GS CHO cells platforms, and as I mentioned, we decide to knock out the glutamine synthetase enzyme, the genes corresponding with this enzyme. We know that once that we block the activity of this enzyme, we will inhibit the productions of glutamine in the cells. Means that these cells now will need from exogenous glutamine to survive. So our first experiment, and this is a representative image, was evaluate the editing efficiency of the pool of these cells. We observed the efficiency around 75%. And the next step was isolate the clones, and then we identify the deletions regarding the genotype. And finally, what we did was establish a sensitivity curve for glutamine, and basically the top clones that we select, first we grow these clones in absence of glutamine. You can see that these clones died. However, in presence of glutamine, the clones survive, expanding as CHO cells wild type, phenotype. So these cells can survive in two ways. First, if we add the exogenous glutamine, or the second way is if we express the glutamines in the cells. Means that we can use this cell line for a selections process instead to use, for example, the antibiotic selection. The second cell line that we have is the clean cut CHO cells double knockout GS-FUT8. And giving a brief introduction about FUT8, FUT8 is an enzyme that can fucosylate the heavy chains of antibodies. In the context of therapeutics antibodies and in cancer cell treatment, what we know is that the afucosylate antibodies will increase the affinity for the receptors in the effector cells, and together will increase the cytotoxicity activity in the treatment, in cancer cell treatment. And here we have a figures in which they compare the afucosylate antibodies versus the wild type, and they evaluate different relations between effectors and target cells. And they observe in all of them a high significant difference when we compare the afucosylate antibodies versus, when they compare the fucosylate antibodies versus the wild type. So taking these information together, what we did was select one specific exon, one exon that when we knock out, we know that we will impact negatively in the activity of FUT8 enzyme. And here is our first experiment. You can see that we achieve around 80% of editing efficiency in the pool of the cells. Then we move on for the isolation of the clones, and we evaluate the genotype of these clones. So here we have a list of top clones in which we identified deletions between 14 VS pairs and 26 VS pairs. And I just keep the clone 1 in which show just the 50% of the indels just for comparison. So our next step was evaluate the food aid gene expression. And what we observed is that for this clone 1, still there is a food aid expression. However, the top clones show absence of food aid gene expression. Moving on for the next experiments, we evaluate the phenotype of these cells. And there are a gold standard assay, which is the LCA staining. Here you can see. So these techniques, basically the LCA is a lectin in which is able to recognize fucosylations in the membranes of the cells. So we evaluate the LCA staining by fluorescence and immunofluorescence. And I'm sorry, it's fluorescence here. And basically, what we detect was in the CHO-GS wild type, the clear staining in the membranes of the cells. However, that was absent in the top clones. Then we evaluate the food aid expressions, and now we use immunofluorescence. And you can see here that we have the expressions in the wild type, and it was absent in the top clones. So together, all these results help us to identify or characterize the double knockout GS food aid. So now moving on for the next technology. So we have the Harboris license-free transposase. And here you can see that we have, in Demeetra, we have a transposons in which we include the glutamine transgenes in order that you can use in combination with the CHO-GS. And we have two versions of this transposase. One is the mRNA, and the second is the protein. And one thing that we would like to highlight is that there is no IP restrictions to use this harboring transposase, and there is no license required. And we can apply it in different fields, like the bioprocessing, cells and gene therapy, and agriculture. So we established GS selections that you can see here. And what we did was transfer the CHO cells, and then during 2 weeks, we select the clone, and we improve the positive populations, you can see here, achieving 100% after selection. We evaluate together the variability of these cells, and what we observed was that the cells were able to expand it, compared with the transposon, which is our control because it's a transient expressions, and the cells died. So as a proof of concept, we isolate clones, and 2 top clones were evaluated to quantify the insertion site. You can see here we have this, the clone 1 that achieved 8 sites, and the clone 2, 12. And we identify the genomic location for them. So finally, we combine this Cas-CLOVER technology together with the harboring transposase, and you can see here that we establish a stable trastuzumab expression cells in which we evaluate the antibody titer. And here we, in the day 14, observe a higher titer and higher cell-specific productivity. So this QP was higher compared with the values observed in the literature. So as I mentioned, we have a protein versions in which we purified this protein. Here you can see the representative image of the transposase together with the complex. And highlighting that advantage to use a protein is that we can work in many types of cells, short life activity, reducing genomic instability, better control of integrations, copy number, reduce the risk of plasmid integrations, and lower risk of insertional mutagenesis, which is safer for cells and gene therapy. So we evaluate the activity of this protein in vitro. This is a classic assay in which basically we know that what is happening is the transposase will pre-complex with the transposons and then will cleave the backbone of the plasmid. So in this gel, we evaluate that. We have the control as the transposon as internal control, here the bands. In the second bands, we have the complex. And finally, we dissociated the transposase using protein K, and then we confirmed that this complex released the cargo, and we confirmed the backbone bands. Together show us the activity of these proteins in vitro. So finally, what we did was using these mRNA versions of protein versions, we evaluate the activity of this transposase as a proof of concept. So we use human T cells isolated from healthy donors, and we use a transposon able to encode it at GFP. And basically, you can see here that we evaluate the GFP, and then we isolate these populations in order to evaluate the copy numbers insertions. In the same way, keeping the same experimental design, we evaluated the protein harboring, and keeping the same idea, we isolate using sorting, and then we evaluate the insertions of these conditions. You can see here that regarding the quantifications of these sites, we identify insertions in node codings and intronic regions than exonic regions, and the same was for the protein harboring. So as a proof of concept, we use a chimeric antigen receptor, anti-CD19, and basically we know that these CAR T cells will recognize CD19 and then eliminate the cancer cells. So we use activated T cells, we transfect with the CAR co-expressing with a GFP under the same promoter. We quantify the GFP, isolate these cells, and we evaluate the pool of these T cells. And again, we show that we have integrations in the genome of these cells, but we didn't find exonic integration. So comparing transposase with lipin, you can see that we have the different versions of using harboring on super piggyback compared with what we have in other companies. And the outperform is higher for us. So in the literature, we observe that there are higher productivity using in between the copy numbers of the cells in 25 and 30. We observe less insertions and higher productivity. So I'm sorry. So one more thing is that we have the license free for harboring. We have some license for super piggyback, but it still is better compared with other information that we have in the literature. So finally, Cas-CLOVER. Regarding Cas-CLOVER, we have a comprehensive freedom to operate cell line development package and service. We know that Demeetra has the license for each component of this Cas-CLOVER. You can see here that we have the IP for the CBC under the University of California, which include the GuideRNA and the Cas9. There is an IP under Helmholtz Munich and Poseida Therapeutic, which is our sister company. And what Demeetra was doing is put all these licenses together in a unique license package, and now we are able to sell for different applications. And also we improving the CLOV51, testing different variants. So summarizing, Cas-CLOVER can be cells in this unique license and also in different application fields like bioprocessing, cell line development, and agriculture. We have the commercial clean cut, choose cell lines, and we have the service to establish cell lines using harboring for example, antibody production. So thank you so much, and for any questions, you can contact our CEO, Jack Crawford. You can contact me or contact our business development manager, Christian Cummins. Thank you so much for your attention.
Thank you very much. We can have a couple of questions before coffee break. There's a fast person.
Hi, beautiful talk. I have many questions, but I'll just ask you a few. I'm a big fan of Cas-CLOVER, but I was wondering, can you multiplex? Can you delete several genes at the same time with this technology? Have you looked for that?
So yeah, so we have this option, right? So we can do this strategy, yes, yes.
And I assume that for making of the guides, the online available biotools are the same because it's essentially Cas9?
Yeah, exactly. So we have an easy way to design, and also because we have the left and right part, we have spacers in which give us flexibility to design and target these specific regions that we have. Yes.
Thanks. And also, just on the slide, it wasn't quite clear to me. Were you transfecting or electroporating actually mRNA into the cells, or were you putting the plasmid in compared to the protein?
Yeah, so no, we use mRNA. Yeah, and we transfect, in our case, is a neotransfection system that we are using for transfection. And yes, it's mRNA.
Great, thank you very much.
Thank you.
Very nice talk. So you have nicely showed that Cas-CLOVER is really better with gene deletions and with knock-ins, which is a feature of the staggered cut, I guess. Because we have also experienced that with like Cas-12 or, for example, MET7, you get like a really higher efficiency for gene excisions since the non-homologous end joining pathway is not as easily accessible to the cell line. Mm-hmm. Have you also tried to use inhibitors of this pathway? For example, some for the DNA PKCs.
No, I think that is interesting. We still haven't tested that. I think that we are more, Cas-CLOVER is more close with Cas9 than the other type of CRISPRs. But I think that this will be interesting for us to improve the applications of Cas-CLOVER.
Yeah, because like the feature of the staggered cut, I think, is like a major advantage that it's producing the staggered cut.
Mm-hmm. Yeah, that is yes, is correct. This is something that we observe. So we have less translocations because of this overhangs end.
Okay, thank you very much.
Thank you.
You decided to use a GS knockout. Yes. But this is an enzyme of CHO cells. Mm-hmm. And there is a reason why my favorite scientist, Mother Nature, put it in there. And one of the things that I know is glutamine synthase can convert glutamate with ammonia into glutamine. Mm-hmm. So it's a rescue pathway to neutralize ammonia in your fat batch. Are you not afraid by deleting glutamine synthase that in the end you might select easier your cells, but that in your fat batch you accumulate ammonia? Ammonia is a weak base which accumulates in acid compartments, which will influence the quality of your material. So are you not afraid for side effects of glutamine synthase knockout?
So I think that this is an interesting point that we are highlighting. I will say, based on my knowledge, this is an established system. And if this is something that is not, is a big risk, I think that probably we will be able to identify because there are...
But is this the reason, is this maybe the reason why we not go up in titers? You know, this is an efficient system, but if it's not limited by the copy number and the site of integration, it might be just inhibited by the side effects of that approach.
Yeah, so I don't know if this really can, I really am not sure if this can really impact negatively in the in these selections.
No, thanks, Cynthia. I'm Jack Crawford. I'm the CEO of Demeetra. These are good questions. We're a gene editing technologies company, so we provide gene editing tools and other like cell lines, platform cell lines to our clients. And GS is a popular cell line. So, you know, for our clients who use GS, they have this opportunity. But we do have a large pipeline of other platform cell lines that we can make, such as a DHFR knockout or a GS DHFR knockout in CHO-K1. So it's basically, we're not too worried about this. If our clients are worried about it, then that's where it happens.
So the same is true for DFHR. You know, DFHR has also a function in the cells. I would go rather for just a plain CHO-K1 and do antibiotic selection, because then you don't influence the whole metabolism in the cell. And right, for gene editing and for selecting the cells, it might not be that important. For fat patches, I know that it is important.
Good, then you can just use Harbor N without knocking out anything in your cell. Yeah.
Thanks. So let's spare the topic of metabolic versus antibiotic selection for the coffee break. And in order to avoid dangerously low caffeine levels in our bloodstream, I propose to finish the session here. Thank you again.